Oh, it's good that you are here. This is the second video, and this one will be going over the second learning target. So I'm hoping that you are finished with your first learning target assignment and your first lab before you begin this. First, I'm going to ask you to open up your digital notebook, turn off your phones, get that space. I'm going to reset my timer here. The last one's about 15 minutes, so that's what I'm trying to get for this one as well. And open up the digital notebook. You're going to start on slide 14. You may have noticed this gummy bear here with no kind of direction. The lab will have direction, so I'm hoping that you've done that already. And on slide 14, it's asking you, how long do you think animals have been on the planet? And it says, my guess, the real number. So take a minute, pause, and put in a guess. What do you think? How long do you think animals have been on the planet? All right, now that you've guessed, let's talk about some things that have happened here along the timeline. So this is billions of years ago, and then you go down here to the 0.7, which is 7 million years, and you come down here to present time. You can see that about 4 billion years ago, you can see the prokaryotes here. The prokaryotes are the ones that have, no nuclei in their cells. And so those are the first signs of life. So life began 4 billion years ago. And if we kind of follow it here, we have some eukaryotes, bacteria, protozoa, still not animals. Sponges are our first real animals. And if you can kind of see where that is right here, you can see that it's about between 0.8 and 0.9. So between 8 and 9 million years ago is when life did evolve, when animals did evolve. And then we have this big explosion of animal life that we talk about a lot in throughout our course, the Cambrian explosion. That was about 5 million years ago, and that was just a huge explosion of animals. Okay? And then if you're interested down here in the hundreds of thousands of years, you can see where we evolved, Homo sapiens. So we are not as old as all these other animals. All right, moving on, you will see target 2, and you will see on slide 15, we've got target 2, okay? I can describe how animals are, 2 R's, related based on evolutionary relationships, okay? This is gonna go over some stuff that you should have covered in biology, but maybe you didn't with the end of the year being screwy, or maybe you just forgot, maybe you fell asleep. Who knows? But it's a good review of evolution because that's how animals are classified. That's how they're named, that's how they're classified. So evolution is change. It is a change in a species over time. And the mechanism of evolution is natural selection. The mechanism, the how do species change, and that is natural selection. Okay, natural selection is the idea that the more fit organism survives the environment. An adaptation is the trait that allows the organism to survive. So the adaptation is a physical trait, whether it be coloration, like in these butterflies, okay? Maybe it's size, maybe it's behavior, but it allows an organism to survive in its environment. So I set up here, it's a change in species. Well, let's talk about what is a species. As adaptations are more and more different from the original population, as organisms change with those adaptations, new species are formed. And so we define a species as a group of organisms that can reproduce and create fertile offspring. And what you see here, what you see here is the liger, okay? And this is a cross between a zebra and a donkey, I believe, and I don't remember the name of that right here. But these, we wouldn't call those a species because these, when you combine 2 species like that, they are actually infertile. So if they are creating infertile species, then they are not, or infertile organisms, then they are not called a species. So we don't call those, just because we're, playing mad scientists and crossbreeding, doesn't mean that we've created a new species. All right, the evidence for evolution. There are 3 pieces of evidence for evolution. I'm just gonna believe that because we're going to talk about these three. There are fossil and structural records, there are molecular records, and there is embryological evidence, okay? Fossil and structural evidence is we look at the fossils like this, or we look at current structures like tailbones. If we have a tailbone, but we don't have tails. So we look at those kind of structures and we see what are the similarities. Molecular records are when we look at DNA, or we can look at proteins, and we line up the A's, T's, G's, and C's, and we see how similar or how different they are. And then embryological evidence, that's when we look at how the embryo develops. So if you remember in the last video, we talked about the blastula folding in and having the mesoderm, ectoderm, and endoderm. We look at after that, when it starts to look like something, okay, maybe not the animal it's supposed to be, when it starts to look at something, that's when we look at what similarities at what stage. The science of classifying organisms is known as taxonomy. And taxonomy, to me, it's kind of boring because it's just kind of like naming organisms or putting organisms in all these different classifications. But it's important that we have a universal system. So it's important that someone here in the United States can talk to someone in Latin America, can talk to someone in Europe, and we are talking about the same organism. And this is very important. They organize them into groups that have biological and evolutionary significance. And I will talk about that in a second here. And why do we need a universal name? Because a common name can be very different. Maybe we call something a red squirrel here, but it's called a brown squirrel in another location. So many of these names are mouthfuls because they're of Greek and Latin origins, and so they're sometimes hard to pronounce, or sometimes just goofy. You may have talked about this in biology, this whole idea of nomenclature. Linnaeus is the one who came up with, and his first name was Carolus Linnaeus, came up with this system of classifications, and it's the kingdom, phylum, class, order, family, genus, species. Okay, so all of these that we talk about, from sponges all the way to the bears, those are all in the kingdom Animalia. Okay, and then if we look at just the chordates, just the vertebrates, that's the phylum Chordata. So we just kind of keep narrowing down until you get to this level here. Binomial nomenclature, if you see the bi, that's two names, is a genus and a species name. And you can see here, this bear, the Ursus arctus, his name Ursus arctus, okay? That is gonna be super important all year long. I'm gonna ask you for scientific names, and that's what I expect. I do not expect one of these other levels. I expect genus and species. So it's important that you understand binomial nomenclature. And the reason that, only the chordates are in this group is because of this point over here. Scientists organize them into groups that are biologically and evolutionarily significant. So they're evolutionarily related. That's why they get put into class Mammalia, or they get put into family Ursidae. Not just because they look alike, because they have an evolutionary significance. Another term, phylogeny, that's studying how the organisms are evolutionarily related. So it's kind of like making a family tree. So that's our next point here of a family tree. Now, how you would make a family tree is you look for characteristics, which when you're talking about phylogeny is called derived characters. Those are just the traits, the qualities, and I have an example in the next slide, that help scientists construct this family tree, which is called a cladogram, right? And let's see a picture of that on the next page here. This is a cladogram. A cladogram is a family tree diagram of a group of organisms. So this happens to be one for the bears, okay? We put it together from the evidence for evolution. So the molecular evidence, the structural evidence, and the embryological evidence. Okay, that's what helps us to create these cladograms. The characters, so not on this one, but on this one, the characters or derived characters here, those are the characteristics: molted external skeleton, segmentation, tiny free swimming. Okay, those are the characteristics, and those are always at the bottom here. Those are always on the line, or in this case, it's a swoosh. I don't know. Okay, so those are always on the bottom. And at the end of the lines are the animal groups. So you can do it by scientific names, or you can do it here by, for example, crabs. The oldest characters and the oldest organisms are always down here at the left side, or at the bottom down here, okay? And it goes from left to right, okay? The ones that have the closest in common are located closest on the cladogram. So here, Ursus tibetus and Ursus americanus, those two are most closely related. And you can see they have the same genus name, okay? That also is an indication that they're closely related. All right, so if you are following along here, you should be on page of slide 17 of your digital notebook, and you should be writing down what the cladogram is. There is an activity here that uses these bears. It's going to open up as a read only, so it's for your information. You should be able to read a cladogram. Probably not going to go over it exclusively, but if you have any questions about it, it's pretty easy. If you have any questions about it, let me know, and we'll address that at one of our discussions. Okay, that's the end of the what is an animal. You can see there's one more slide. That's our last target, so that'll be a separate video altogether.